This is our introductory lecture of this course. In this lecture, you will be introduced with a few terms like gene structure, coding sequence, HapMap project, and ensemble database. You might be familiar with these terms, but this is very important to revise the basic concept of these terminologies before initiating the research work involving the in silico tools of this course. As we know that a gene comprises of the exons as well as the intronic regions between the start codon and the stop codon. You can see here the regions upstream the start codon and also some regions which are downstream the stop codon, which are labeled as regulatory sequences. The upstream regulatory sequence is comprised of enhancer, promoter, and 5' UTR. Basically, these regulatory sequences control the level of expression of structural genes, and the enhancer are the DNA sequences which are which increase the expression of a gene when they are bound by transcription factors, and the promoter is the binding site for transcriptional machinery. While the downstream regulatory sequences are comprised of 3' UTR and silencer. Silencer are the DNA sequences which are capable of binding the transcription factors called repressors, and they reduce the expression of a gene. 5' UTRs and 3' UTRs. These, as their name indicate, they are the untranslated regions. These regions are transcribed into the messenger RNA, but they are never translated into a protein. The 5' UTR is critical for ribosome recruitment to the messenger RNA and start codon selection, and it plays role in shaping the cellular proteome. While 3' UTR is the part of messenger RNA that immediately follows the translation termination codon. And you can see here the open reading frame, which is the region of the gene between the start and the stop codon, and it is comprised of the exons as well as the introns. Now, when the DNA is transcribed into the pre-messenger RNA, it is comprised of 5' UTR as well as the 3' UTR and the exonic and the intronic regions. Then it is modified via splicing, and we get a sequence of the gene in which there is concatenated exon sequence. Like there are no more introns between the exons. The 5' UTR is still present, and 3' UTR region is also present, but there are no introns. And this region in this part of messenger RNA, which is without introns and is capped at 5' end while tailed at 3' end, is known as mature messenger RNA. So the region between the start codon and the stop codon in the mature messenger RNA, in which there are no introns and only exons exist, this part is known as coding region, which is directly translated into the proteins. Now, in order to apply the tools on for characterization of any gene, you are supposed to retrieve two types of data regarding a gene from the different genome browsers like Ensemble, UCSC Genome Browser, NCBI, or JBrowse. These two data are the coding sequence or the open reading frame of the gene, as well as the single nucleotide polymorphisms of the gene. As we know that if we are able to access the coding sequence of the gene from these different databases, then this is the contribution of the Human Genome Project. Similarly, another project known as HapMap project was initiated in order to determine the genetic variations among the human population of Asian, African, and European ancestry, And these genetic variations might affect the health and disease of human. DNA sequences of any two people are 99.5% identical, and like there are very small amount of variations, but these variations greatly affect an individual's disease risk. The sites in the DNA sequences where individuals differ at a single DNA base are called single nucleotide polymorphisms. And sets of the closely located SNPs on the same chromosome are always inherited together from the parent to the offsprings. And this pattern of SNPs, which is located in the form of a group or in the form of a block, is known as a haplotype. And these blocks may contain a large number of SNPs, but a few SNPs are enough to uniquely identify the haplotypes in a block. The HapMap project actually is presenting a map of these haplotype blocks, and the specific SNPs that identify these haplotypes are called as tag SNPs. Now, you can use any of the database like UCSC or NCBI or Ensemble to retrieve these two kinds of data about your gene. But as per my experience, the Ensemble database is more easy to access the SNPs, and also it is presenting the data in a very comprehensive way. When you will use this database to retrieve the data, then you will also experience this fact, and we will also compare the same data which is presented on the Ensemble database and on the NCBI. So you will also find easy to use this database. Therefore, I am also teaching you this one. And Ensemble Genome Database Project is a scientific project at the European Bioinformatics Institute, a well-known genome browser for the retrieval of genomic information. This is the homepage of Ensemble database. And in my next video lecture, we will see how can we retrieve the gene coding sequence and the SNPs from this database. Thank you so much.